Yeah. Adult penis. Okay, so downward we have the ventral side, upward we have the dorsal side, and ventrally we have this kind of tissue called corpus spongiosum, into which the urethra is embedded. Because seemingly this urethra is embedded into the corpus spongiosum, this is the spongy part of the urethra, or also called penile part. And the lining epithelium here is characteristically stratified columnar. Inside this epithelium, there are endoepithelial glands, which are pale-stained cell masses, multicellular glands, because they are mucus-producing glands. Mucin was lost during formalin fixation. And they are called endoepithelial because they are inside the lining epithelium, in this case inside the lining epithelium of the male urethra. This corpus spongiosum is a kind of tissue which is a mixture of connective tissue and smooth muscle. Here you can see smooth muscle cell nuclei in cross-section, for example. This is smooth muscle cell nucleus in longitudinal view, or this one is even better. And this is collagen fiber, obviously. This is probably a fibrocyte, or so it's a mixture of smooth muscle and connective tissue. Inside this corpus spongiosum there are irregular cavities lined by endothelial cells. These are the cavernae, special blood vessels. Cavernae. Caverna is the singular, cavernae ending with AE is the plural form. I don't understand. Gland. Endothelium, like any blood vessels. Yeah. Now, yeah.
There's these glands that there's ducts from the urethra go to.
You mean this?
Yes, the small. Yeah, exactly. I don't really understand what's their purpose.
Such a structure is probably a paraurethral duct. Have you heard about them? I drew it.
Not with us, not with our group.
Oh, not. So this is a longitudinal view of the male urethra, right? This is where the urine is flowing downward. And these are blindly ending branches of the urethra, which are running more or less parallel with the urethra. That's why they are called paraurethral ducts. The point is that they are ending blindly, and this blind end is always pointing upwards. And the clinical significance that because of this kind of anatomical arrangement of the paraurethral duct, like these, bacteria may reside here and proliferate for a long period of time and may cause inflammation. Because the urine cannot really wash these bacteria out of this territory, that's the problem. And I believe that this is one example for such a paraurethral duct. Inside the lining epithelium of the paraurethral duct, you may also find endoepithelial glands, which may therefore be termed as paraurethral glands.
But what is the reason that we have those glands, the paraurethral?
I cannot give you the reason. This is something like why humans develop this way and not some other way.
Okay, fair enough.
We just need to accept that somehow during evolution our body appeared like this. In some cases we can argue that this is a good arrangement biologically because of this or because of that. Here we cannot argue why this is a good arrangement for us. It is actually disadvantageous rather than advantageous according to our understanding. So biology, so you cannot always account for the reason why structures develop like that. That's the problem. And this is one of those. Now the other thing, we have these two structures, this one and that one, called corpora cavernosa, or cavernous bodies, which are surrounded by a dense connective tissue called tunica albuginea. Tunica albuginea, very rich in collagen fibers. And between the two corpora cavernosa, this continuation of the tunica albuginea is called septum penis. Septum penis. And in these corpora cavernosa, you can see the cavernae. So this is actually the origin of the name. And during erection, these cavernae are filled up by blood. That's why the penis gets erected and more rigid. And during erection, these cavernae receive blood primarily from this artery called deep artery in the middle of the corpus cavernosum, deep artery. During the flaccid state, so when the penis is not erected, blood supply of cavernae is from elsewhere, from here, from these so-called dorsal arteries, which is paired. Dorsal arteries. But during erection, the major source of blood is this deep artery. And during erection, this corpus cavernosum is enlarging. That's why the penis is enlarging. And the venous outflow from these cavernae is at the periphery. But because of the enlargement of the corpus cavernosum during erection, during which the cavernae are getting filled up by blood, so the corpus cavernosum tends to enlarge, but because the tunica albuginea around them is rich in collagen fibers which are not elastic, therefore the peripheral part of the corpus cavernosum will get compressed against the tunica albuginea, compressing the veins at the periphery, which are responsible for the drainage of blood. So blood cannot flow out, but still arriving through the deep artery. So this is the key to erection, and is very much related to the high collagen fiber content in the tunica albuginea, because if the tunica albuginea was rich in elastic fibers instead, elastic fibers would allow expansion of the tunica albuginea, venous outflow wouldn't be blocked, and erection wouldn't occur. But in contrast, around the corpus spongiosum we also have kind of dense connective tissue, which is fiber rich. But here the fiber composition is somewhat different. More elastic fibers are present. And that's again important functionally, because during erection, corpus spongiosum is also enlarging because cavernae are here. But this enlargement is not blocked by the surrounding fiber-rich tissue, because elastic fibers allow expansion of the corpus spongiosum. And the biological significance that this way, during ejaculation, the urethra will not be compressed. So the semen can flow through. So as you can see, the difference in fiber composition in the tunica albuginea around the corpora cavernosa and the connective tissue which surrounds the corpus spongiosum is highly important for survival of humankind. Because it would be, if the fiber composition in these two territories is reserved, then either erection, so erection wouldn't occur and semen wouldn't be able to leave the urethra. Both are against maintenance of the species. So such a simple histological feature, it can be very, very important in certain cases. Yeah.
Any diseases that you do in reality the development that we need to know for this?
Well, obviously one thing at least what you need to know, because it is illustrated in the other section here, number 17, which is an embryonic or fetal penis. This phimosis, which it's not really a malformation, but may cause problem. So this is a fetal penis in cross-section. Here is the urethra with the corpus spongiosum and with the two corpora cavernosa. Here you can also see the tunica albuginea and the septum penis, male urethra, and in addition to these endoepithelial glands, like here, this paler territory, or like that, here, endoepithelial glands. In this case, you can see extra epithelial glands, like this. These are outside of the lining epithelium. These are also mucous glands within the corpus spongiosum. These are the glands of litter, exoepithelial glands. It's an osseous name. Now, so regarding the phimosis, phimosis is here and also here. These are territories where the surface of the developing glans, which is here, still not got separated from the inner surface of the prepucium. This is the prepucium, which is a skin fold, which can at least partially cover the glans. So this is physiological phimosis. Physiological because at this time of development it is still normal to have this fusion between the glans and the prepucium. But this fusion has to disappear by the time of the start of the puberty at the latest, by the time the boy is having his first erection. Because if this fusion remains and the first erection occurs, then the prepucium cannot be retracted from the surface of the glans, which should normally happen during erection. If it cannot happen, it causes pain. So if it is not resolved spontaneously by the time of the start of puberty, then a small surgery is needed to cut this fusion. Here in this territory, this fusion already disappeared. Okay. And here is the prepucium. This is the outer surface with the stratified squamous keratinizing epithelium. And here is the inner surface of the prepucium with the developing stratified squamous keratinizing epithelium. And here is the third developing epithelium, which is the stratified squamous keratinizing epithelium of the glans. At the territory where the phimosis still exists, like here, in this territory, at the territory of the fusion, what we can see is nothing more than here one stratum basale, one row of cuboidal cells, and another row of cuboidal cells, another stratum basale. And in between the two stratum basale, we have a unified stratum spinosum. Other layers haven't appeared. Okay, so this stratum basale will be the stratum basale of the epidermis of the inner surface of the prepucium. This stratum basale will be the stratum basale of the epithelium of the glans later. Here in the prepuce, you can see a very fine bundle of smooth muscle cells here. This is called dartos tunic. This smooth muscle bundle is continuous with the smooth muscle bundle of the scrotum. And this dartos tunic separates the prepuce into two parts. Two fascia. The upper part is the fascia superficialis penis, superficial fascia, and below we have the deep fascia. Actually, these are dermis, a unified dermis layer of the skins, which is the outer one and the inner one. And because this is skin here, not a surprise that we have developing sweat glands like this, also here or there. And developing sebaceous gland here. Yeah. No, this is not a hair follicle, this is just a sebaceous gland. Well, I wouldn't say no. It's a sebaceous gland. Yeah, so this is the problem what you spotted. That this is said to be thin skin, but without really hair follicles. Biological nomenclature. Hate it. Yeah.
I think Christina told us last lesson about this, that there's a muscle
Muscle.
that blocks the passage of blood normally.
Blocks what?
Blocks the passage of blood towards the penis. And whenever it's innervated, it relaxes the muscle. And
Oh, maybe you are referring to the Abner's cushions,
what?
don't you? Abner's cushions. Here in the corpus cavernosum, you can see structures like something like this, a kind of evagination towards the lumen of the caverna. And this fibromuscular tissue contains smooth muscle cells as well. So the contractile state of these smooth muscle cells in these territories, which are called Abner's cushions, is related to the fact, to the extent these structures are evaginating towards the lumen. And it is believed that such Abner's cushions this way can regulate blood flow inside the cavernae,
So
especially during erection.
are they always like extended, let's say, like the muscles?
No, not always. This is variable because during erection, nitrogen monoxide is liberated as a substance, and it causes relaxation of muscle. And this is the basis, the biochemical basis of the pill called Viagra, which is given to males who have difficulty with maintaining erection, because eventually it will trigger this kind of biochemical pathway.
So this relaxes these muscles?
Yeah. Okay, so this is another aspect of the story. And here in the adult penis, I can show you something else, like this. Can you recognize?
Pacini itself?
Yes, this is a Pacinian corpuscle, which is a mechanoreceptor. Again, important, because otherwise males wouldn't feel too much during sexual intercourse without these mechanoreceptors. Okay? So again, a tiny microscopic structure which is related to survival of humankind. And here are nerves beside which conduct the sensory information, obviously. Yeah.
These are ganglia? Or cells?
No.
What's the difference? Like, how do I see the difference between the ganglia and just nervous cells? Maybe a stupid
A
question.
ganglion is a collection of ganglion cells and nerve fibers. Well, this is first semester material.
I know, maybe, yeah.
Yeah. One more thing here, dorsally, a piece of the skin remained, and also here you can see the arrangement of certain vessels. It's a rather unusual arrangement. Here is the dorsal vein in the midline, and on the sides there are two dorsal arteries. And even more laterally, here there are dorsal nerves, nerve fibers. Yeah. Well, by just looking at them, we cannot tell. Both sets of nerves are present. I already showed one.
No, I mean, the person presenting has both, so the loss between
We don't really use this terminology in relation to the Pacinian corpus, so we can talk about sensory nerves there. Okay, so what should we talk about next?
Could you talk about the urinary bladder slide 64 and 2?
This one? No. Nothing too much special here relative to the other urinary bladder slides. This is just a thinner-walled urinary bladder, which may have two different reasons why the wall is thinner here. One is natural biological variability, but maybe this is a too big variability because this wall is, I would say, at least 5 times thicker than this one, if magnifications are identical, but I don't know. But another possibility that if somebody keeps urine without going to toilet for longer period of times, and this is a kind of behavioral pattern, it may increase chances for having a thicker wall in the urinary bladder. So we don't know. I changed the orientation because this way we have the lining epithelium facing upwards. So obviously urothelium, you can see umbrella cells, sometimes binucleated like here. And below the row of piriform cells resembling the shape of pears, the fruit. And here you can see the lamina propria, no muscularis mucosae, no submucosa. Although, if you look at this lamina propria, you can easily realize that this upper portion of this connective tissue is looking different than this lower portion. So the color is different. So that's why in certain cases, in certain books, the books still use the term submucosa, although there is no structure which would separate the connective tissue into two parts. But the appearance is visibly different in the upper and lower parts. So based on this observation, some books still say that this upper part is the lamina propria and the lower part is the submucosa, but I emphasize no muscularis mucosae in between. And again, during exams you need to emphasize that the starting point is that in the urinary system there is no muscularis mucosae and no submucosa. And then you can refine the statement. So, just for simplicity, I just talk about a unified lamina propria, which is followed by an external muscle layer, entirely smooth muscle, where three differently oriented smooth muscle layer can be distinguished. And then the outermost tissue layer is either adventitia or subserosa and serosa, because part of the urinary bladder is covered by peritoneum, part of it doesn't. Here, well, I'm not sure that these might represent mesothelial cells. If they are, then we can conclude that this is the serosa layer, and here the loose connective tissue is the subserosa. And at least in this part, the bladder had a peritoneal coverage. It's an infraperitoneal organ, so the apex is covered by peritoneum. And the extent to which it is covered, it is dependent also the fullness of the bladder. Yeah.
Regarding the smallest what defines
in the sections, in our urinary bladder sections, muscularis mucosae is not visible. We have 3 urinary bladder sections. As far as I know, none of them contains muscularis mucosae.
In general, like all the
Oh, so the general description should be that in the territory of the trigonum, we never have muscularis mucosae. Outside the trigone, we might find traces of the muscularis mucosae anywhere outside the trigone. But in all sections, we cannot see muscularis mucosae.
First is sections from the trigone region, so you definitely have adventitia.
Yes, because that's the base, so it is not covered by peritoneum. Anything else? Yes?
Can you explain the transition between the urothelium to the stratified columnar in male?
Explain the urothelium.
The transition from the urothelium to the stratified columnar.
Or the transition. I'm not sure what you mean. What I talked about earlier when we dealt with the urothelium.
No, sir, it's in the male urethra.
In the male urethra. Well, urothelium is urothelium, independently whether it is in the urinary bladder or in the male urethra. I still don't understand the question.
Like the transformation between the two types of epithelium.
The transition,
Yeah.
not transformation. Well, I'm still not sure what you mean, but do you mean that in the fetal penis, the urethra shows signs of transition in terms of the lining epithelium? Okay. So if you look at this fetal penis, the lining epithelium of the urethra here, in some places like here, looks like stratified squamous non-keratinizing. You can see squamous nuclei on the surface. But in some other places, let's say, let's say here, or here, the epithelium is very much looking like stratified squamous columnar epithelium, because on the surface you can see columnar nuclei. And therefore we can conclude that because both types of epithelium is visible here, this is a kind of transition between the spongy part of the urethra and the navicular fossa. Navicular fossa is the most distal part of the male urethra, located in the distal part of the glans penis. That's a widened part. And the typical lining epithelium in the navicular fossa is stratified squamous non-keratinizing. But in the spongy part, the typical lining epithelium is stratified columnar. So because both of these types are visible, we can propose that this is a kind of transition. So the section is probably from the proximal part of the glans where this transition occurs. So this is what you wanted, okay. Yeah.
Also, the fossa is ectodermal, right? The epithelial fossa is ectodermal.
You know, that's a transition, as the type of the epithelium suggests. So that's a transition there. But development of genitalia is coming. Okay, so let's return to that, because we will have a seminar dedicated to this later. Okay. Maybe one final question, we have some time. No? So in that case, please notify your group mates who are not here that